Before we get started with the practical session of single-cell ATAC-Seq data analysis, first of all, we will discuss the theoretical part of single-cell ATAC-Seq data analysis and the working principle behind this analysis. So before going into a deeper detail, first of all, let me tell you that what a single-cell ATAC-Seq data analysis is and what it does. Single-cell ATAC-Seq data analysis is a sequencing technique used to measure the chromatin accessibility at single-cell resolution. It was developed by Borenstro et al. And the paper was published in Nature Method in 2013. Single-cell ATAC-Seq data identify open or active regulatory regions in individual cells. It reveals cell type specific regulatory landscapes, and it complements single-cell RNA-seq data by explaining why genes are active. So the key question that it addresses is which DNA regions are accessible in each cell and how does this define the cell identity. That is the overall experimental workflow of single-cell ATAC-Seq data analysis. First of all, individual cells or nuclei are inserted with the TN5 enzymes that introduce ATACs into the open chromatin regions. Let me tell you one thing, that TN5 enzyme cannot introduce ATACs into the closed chromatin because that is highly packed, and the TN5 enzyme cannot access the DNA part of that region. So it only introduces ATACs into the open chromatin regions, and after introducing ATACs into the open chromatin region, all the fragments generated from the open chromatin regions are then proceeded into the sequencing machine. Here, individual cells or nuclei are isolated, and the TN5 enzyme inserts sequencing adapters into the open chromatin regions. These fragments are then sequenced and computationally analyzed to identify accessible DNA region across thousands of single cells. And finally, cells are clustered based on their chromatin accessibility profile and visualized using dimensionality reduction method such as UMAP. Each cluster represents a group of cells with similar regulatory landscape and corresponding to distinct cell types or states.